Hey, Assalamu alaikum. In this video, we will understand the toxicology topic, snake antivenom production, and the synthesis as a vaccine. So, what is the snake venom? We will understand first, and we need to understand. So, a venom is important for predation and capture prey easily from the snake. So, snake will bite the prey to capture easily and kill easily to digest and a lot more things because it contains enzymes so the digestion possible. This snake will release the venom through their teeth. Teeth contain a inside is the hollow tube mid of the each teeth. So both teeth um, contain a duct. So here is the venom gland which that's contain a venom release from the teeth contain a venom duct. Oh sorry, glandular duct or gland duct. So venom basically contain proteins plus enzyme. So basically enzyme is also made up of protein. So you can say that is the whole protein. Destroy our cell. So it will destroy our cell. We can separate the venom through the some mechanism, the venom collection. So in this the venom basically uh, uh, basically is the anti-venom is self-saving treatment for snake bite and venoming and preparation is complex multi-step process, but we will understand the overview in this video. So here is the whenever you can use the technique to how to take the venom from the gland of the snake, venom gland. So venom collection basically carefully selected through some jar or some any type of basically beaker which the carefully selected and milked for their venom and stimulating glands of venom. The stimulating through the bite, so you can put the teeth of the snake in the adjacent side to remove the venom. So the number two is the collection venom is then carefully diluted and standardized to ensure consistent batches for immunization, and small non-lethal doses will be separate to inject the animal horse, and in this the animal immunization. In this the horse whenever the inject these small non-lethal dose into the horse and in this way enter into the blood circulatory system of the horse. So this is the blood circulatory system of the horse contain RBC and WBC. This is the T helper cell. So other animal can donkey, goat, llamas and camel also use for this purpose. So remember about that things. Not only horse. So these whenever inject these small non-lethal doses, here is the antigen. Because this is the antigen and the animal body will recognize as an antigen and activate the T helper cell. This T helper cell, and here is the macrophage and dendritic cell, also inhibit this antigen. But after the inhibition of this antigen through lymphatic system, recognition through a T helper cell, and T helper cell will recognize to help to activate the naive B cell to proliferate into the plasma cell and B memory cell. This proliferation of the plasma cell and B memory cell will basically quickly production of antibodies from the plasma cell, but the long term throughout the life of the animal the B memory cell will remain active. Whenever the infection will occur, will lead to suddenly and quickly produce the antibodies for combating the pathogen like the small or big venom dose. So here is the fourth step is the antibodies harvesting system. In this the collection of blood after week from the horse, and that collection of the blood will separation of plasma, and plasma contain basically is known as serum. Serum is the plasma. So remember water color containing serum, which that contain albumin protein and antibodies, and in this way it should be separate, and in this way the centrifugation or other process will inoculate the serum, and the serum will be separate, while the RBC, WBC and platelets will be separate and remain down of the sample. So this is the blood. So in this, the albumin and antibodies and mineral plus water will be separate, and separation of plasma. After the separation of plasma basically contain antibodies, which that is very important, and that is against the venom of the snake. So in this way, the purification and standardization remove the impurity. Serum protein can cause adverse reaction in patient, so we should carefully separate. And purification method is very important. Purification method can be salting out, enzymatic digestion, chromatography, and filtration. And the sixth step is the testing and packaging. Testing means we should use the animal after the bite of the snake. The patient inject this serum containing antibodies to sudden cure. So remember, the testing and packaging is very important. The testing is either helpful or not, this production and this vaccine, which that is very important after the testing and packaging. So basically is the company will give to the patient, and basically is the complete process will occur, and in this way the anti venom will be synthesis.